I think we are going live. There is always a bit of delay, like 20 seconds for us and the YouTube. We've been working out this as well. All right, I just heard from my mom that she's going to take some vaccination, and we are.
Oh, on. That's great.
That's great. Yeah, and we are on now. So welcome everyone to our last lecture of this graduate course on cell death and the immune system. It is my great pleasure to introduce the next speaker. Doug Green is the Peter Doherty Endowed Chair in Immunology and the Chair of the Department of Immunology and the Cancer Center Program Co-Director at the St. Jude Children's Research Hospital in Memphis. But to me, Doug, much more than Doug, you are a dear friend, an all-time mentor, and someone I cannot thank enough for everything that you've been doing on my behalf. Actually, it's fair to say that the most enjoyable time in science to me was the 3 years I spent in his lab together with his buddy here, Seamus Martowicz, who gave the first lecture of the course as well. So this whole course has been really a great pleasure to me. And I'd like to take this opportunity to thank all the friends that agreed to accept our invitation to share their experience and knowledge with all of us. Doug has committed himself to the study of mechanisms of cell death and survival since the late 80s. His contribution to the field is absolutely immense. I will not mention any particular paper among over 130 that I could count, which has received more than 100,000 citations. The age factor is currently 159. Suffice to say that among numerous honors and awards, Doug is on the ISI list of the highly cited scientists in immunology since 2003, and in molecular biology since 2014, and was recognized by the Web of Science in 2020 as one of the most cited scientists of all time. Doug is a distinguished fellow of the American Association of Immunologists, member of the National Academy of Science in the US, foreign fellow of the Royal Society of Canada, and I could be going for another half an hour talking about his awards and so on. So I'm just going to stop here and thank you very much again, Doug, for sparing your time, your precious time here to our course. Thank you.
Thanks, Gustavo. It is such a pleasure. The only huge regret is that we're not all together in Sao Paulo.
That's right.
That's maybe next year, I hope.
Definitely.
I hope. So thanks so much. And I hope this has been a really good day for people. People are learning something about cell death. You're hearing from some of the great cell death researchers of the world today, which is really phenomenal. And you've met a lot of my friends, which is also great. So to wrap this up, I want to look forward and not backwards, really to see what do the next 10 or 15 or 20 years of cell death research look like, maybe. And to do that, I thought we would do it in terms of riddles in the dark, because we don't know the answers to the questions that I'm going to put out there, but I'm going to throw out some teasers, some little bits of information that have been coming out either over the last couple of years or new unpublished things, to really ask 5 riddles about cell death, things that we really don't know. So just to start, and let's see if it'll actually move my slides forward. Hopefully, yep, here we go. Woody Allen said, "My relationship with death remains the same. I am strongly against it." Those of us who are speaking today, we're not so against it because cell death is really important for biology. But we can also understand why he would say he's against it. Here's a little factoid. In the 1970s to 1980s, in that decade, very, very few papers on cell death. This is the percentage of all publications in biomedicine. By 1990, there were a few papers, and then the field exploded until by the decade from 2000 to 2010, more than 2.5% of all published papers referred to some form of cell death in the publication. And in the last decade, that increased almost 3%. And although it's no longer exponential growth, it continues to rise. Cell death is a fundamental biological property, something that we think we understand to some extent, but as you'll see, there are so many things we still don't know. So, riddle number 1, how deadly is death? I'm going to start to approach this question. Really the question is, when you engage a cell death pathway, are you dead if you're a cell? And I'll start with not looking at apoptosis because we'll come to that later, but I want to start with another form of cell death, necroptosis. And I suspect that you've already become familiar with this cell death pathway through the other talks that were presented today. But just to remind you that necroptosis is induced by a number of different receptors. Toll-like receptors can engage it through the adaptor TRIF. The TNF receptor 1 and other death receptors can engage this through the engagement of RIP kinase 1. And another receptor for a nucleic acid receptor called ZPP1 can also engage this. And all of these converge on RIP kinase 3 and its activation. Now that activation is countered by a caspase, caspase 8, especially caspase 8 when it's in a heterodimer with C-flip. The enzymatic activity of caspase 8 C-flip destroys RIP kinase 1 or RIP kinase 3 and prevents this process from occurring. When RIP kinase 3 is activated, it phosphorylates a target molecule, MLKL, and that forms openings, disruption of the plasma membrane, and the cell dies. Oh, sorry. When you engage the phosphorylation of MLKL, it really kills the cell. Here's a 3-dimensional scanning electron micrograph reconstruction. That's the intact cell. That's what the intact cells look like. When MLKL is activated, the plasma membrane is destroyed. So you would say that activation of necroptosis is absolute death of the cell. But if you notice, intracellular membranes stayed intact, and that's going to be important. So I'm going to start off with some recent discoveries from an alumnus of our lab, Andrew Oberst, who started these experiments when he was still in my lab several years ago and published them in 2019. And what Andrew noticed using a system where he could activate RIP kinase 3 directly by inducing its oligomerization, something he developed in our lab 10 years ago, that you express this form of RIP kinase 3, you can add a dimerizer agent, it'll activate the RIP kinase 3 and induce that cell death that you just saw in the last images. But it also induces the production of cytokines from the cell. And this is through a complex that Seamus Martin described called the fatosome, which can engage NF-kappa B activation. The cells die, but they also produce cytokines, in this case, interleukin-6. And what you can see is that here where the arrow is, the cells are dead, but they seem to still be producing cytokine. If Andrew added an inhibitor of transcription, actinomycin D, that didn't affect the production of the cytokine. That continued to be produced after all the cells appeared to be dead. But if he added an inhibitor of protein synthesis, there's my pointer, the production of cytokines stopped. To look at that in a little more detail, he and his graduate student separated the dead cells and the supernatant. And if he incubated those corpses, they continued to produce cytokine. They were actually making the cytokine. So a way that you can look at protein synthesis is by the incorporation of puromycin. This blocks protein synthesis, but the puromycin actually gets integrated into the nascent peptide that's being generated from ribosomes. And there are antibodies that can detect the puromycin. If you stain a cell with puromycin, the proteins that are being synthesized stain and you get diffuse cellular staining. If you look for endoplasmic reticulum, some of that protein is, of course, being produced in the endoplasmic reticulum, but much of it is being produced in the cytosol. If you add cyclohexamide, all of that signal goes away as expected. But when he killed the cells, when he took cells that were now necroptotic because they'd activated RIP kinase 3, he could still see puromycin signal that co-localized with the endoplasmic reticulum. And that was blocked by cyclohexamide. These dead cells are still making protein in the endoplasmic reticulum. And just like in the images I showed before, but these are other EMs, endoplasmic reticulum in the dead cells, these are fully necrotic cells, endoplasmic reticulum is intact and it still has ribosomes associated with it. If he induces the cells to die by the activation of RIP kinase 3, they still continue to produce cytokine that's blocked by adding an inhibitor of protein synthesis, but it's also blocked by permeabilizing now the endoplasmic reticulum with digitonin or by preventing Golgi function for secretion. The secretion apparatus is still intact in these dead cells. And that might be important because another thing that he showed in a different paper, also published in the same year, is that if you introduce an inducer of necroptosis, now using a RIP kinase 3 that spontaneously activates, if he expresses this in a tumor, those tumors now don't grow, and neither do tumors that are not injected but are in the same animal. He's inducing an immune response against the tumor by the induction of necroptosis. And that also happens if he introduces already dead cells into the tumor. So if he introduces necroptotic cells into the tumor, they will induce an immune response, the activation of T cells specific for an antigen associated with the tumor. And if he blocks protein expression in those already dead cells, that effect goes away. So even a dead cell can still be biologically active, at least in this form of cell death. Now there's more to it, because if the cells can engage the cell death machinery, when are they actually dead? And that question, again, thinking about necroptosis, we'll come to apoptosis later, was addressed by this guy, Yinan Gong, who left the lab a few years ago, talking about experiments that he did more than 3 years ago. Again, using the same kind of system that Andrew developed, inducing necroptosis by inducing the oligomerization of RIP kinase 3 or directly inducing the oligomerization of MLKL. So if either of these are dimerized, they induce the death by necroptosis. One of the things that Yinan discovered, and this is important for for all of us studying cell death, is that if you induce necroptosis in cells, they externalize phosphatidylserine, just like in apoptosis. This does not depend on the same mechanisms as in apoptosis. It's not caspase dependent. It's not dependent on the scramblases that are activated during apoptosis, but phosphatidylserine is exposed on the cell surface before the plasma membrane loses its integrity. So if you remember, we can stain with annexin 5 and see staining of the cells before they'll take up, for example, propydium iodide or 7-AAD. Many people say that the exposure of phosphatidylserine or the binding of annexin 5, and by the way, as a little aside, Seamus Martin, who's here with you and spoke to you today, is the one who discovered that annexin 5 is a universal indicator of an apoptotic cell. Annexin 5 binding before loss of plasma membrane integrity is a marker of apoptosis. It was only much later that Yinan discovered that the same thing applies to necroptosis. But he could use that now to isolate cells that have activated the MLKL but had not yet lost plasma membrane integrity. So these are not the corpses that Andrew studied. These are cells that are on their way to being corpses. They've activated MLKL and they are in the process of dying. As I said, he activates RIP kinase 3 or MLKL by the addition of a dimerizer. The dimerizer has two parts to it that bind each of these dimerization domains, but he could also separate those dimers by adding a monomeric so-called washout agent. So this is a way that he can kill the cells, but while they're in the process of dying, inactivate that signal. So he takes those annexin 5 positive cells. They will all progress now to being fully dead. They'll lose their plasma membrane integrity. That's on the left. This is with a mock reagent that doesn't disrupt the RIP kinase 3 dimer. But if he takes those annexin 5 positive cells that have activated MLKL, here's the phospho-MLKL that's present in those cells, and now blocks the, dissociates the RIP kinase 3 so it becomes inactive, many of those cells return to being annexin 5 negative with intact plasma membrane, and those cells grow and survive. We call that resuscitation. Don't have to do it with those artificial systems. If he induces necroptosis by engaging TNF receptor in the presence of a caspase inhibitor, this is in a human cell line. Again, he can isolate those annexin 5 positive cells. They all progress to dying. But if he now blocks MLKL with an inhibitor, necrosulfonamide, in those cells that have already activated MLKL and are in the process of dying, many of those cells now recover, survive, and can proliferate, as you can see here. Interestingly, if he takes those cells that have exposed annexin 5, but still have the potential to live, and compares them to apoptotic cells that also can be recovered. This is from a database from so-called anastasis, and we'll come back to that later. Changes in gene expression in cells that have activated death pathways but might survive. There are differences between the cells with the potential to survive apoptosis and cells that have the potential to survive necroptosis. Sorry, these are the survivors of necroptosis. These are the survivors of apoptosis. These are the gene differences, but many of the genes that are induced in surviving cells are similar. And that is a whole area of research that's unexplored. How do cells survive the engagement of a cell death pathway? So how deadly is death? Even when a cell is dead, it can continue to perform some functions. And cells that engage cell death pathways can be resuscitated. This may happen under physiological or pathological conditions. We don't know the extent to which this occurs in vivo, but what are the implications, for example, in cancer, and how can we find out? How can we identify a cell that has engaged a cell death pathway, but goes on to live? And what are the consequences? And we'll come back to that in a completely different context later. But that's the first riddle. I'll stop here for a moment if there's a question or two.
Doug, I think there's a question from April Darcy. What sort of proteins are the dead cell ER producing? Is that some particular or every single one that they are already? You know, is there any?
So in Andrew's paper, he mainly interrogated cytokines because these were important in the anti-cancer effects. But we suspect that all the proteins that are being synthesized for secretion or membrane expression continue to be made, at least for many hours after the plasma membrane integrity is lost.
All right. Thank you.
Can I ask a question?
Of course.
So, Doug, are the cell death assays too sensitive in a way, so that cells that are being marked as dead aren't really dead in metabolic terms? That's one question.
I think the answer to that, well, what do we mean by too sensitive? They're sensitive enough that we can detect that the cell death pathway was engaged, but they may not be sensitive enough to tell us that that cell is actually dead. And our definition of when a cell is actually dead may have to be modified. Yeah.
And maybe you mentioned that, but do inclusion of caspase inhibitors in those experiments affect the kind of rate of resuscitation in any way?
No, not at all. So at least in the necroptosis experiments, they are caspase independent. Yeah. But, go ahead.
Sorry. It's a bit noisy here in my home, but is there any correlation between that the protein that Vishwa described, means one with this cell death, really death?
Is there a marker that can tell us that the cell is really dead? And I think, I suspect that it will depend on what biological activity you're interested in. So if you're interested in whether that cell can make another cell, I think we're pretty satisfied that once a cell engages a cell death pathway, it will reach a point where it can no longer generate the biomaterials to make another cell, but eventually will lose all activity. You know, think about it in terms of when we think about death of an organism, an individual can be brain dead, but still have biological function, or organs can survive, or as we know, cells can survive. And so our definition of death will depend very much on the biological outcome that we want to read out. So if we're interested in when does a dead cell stop making cytokines, that might be very different from when the cell loses its plasma membrane integrity. If we're interested in metabolic activity or other processes that may go on in the cell, those may go on much longer than when the cell's heart stops, right? And what are the biological consequences of that? That's the question. And like I say, these are riddles. These are things that I think we'll explore in the coming years. So let's go on to riddle number 2. And this is a little different. So first we were talking about when a cell is dead or is on its way to die, when is it dead? But there's another kind of question. When is a toxin, when is something that kills a cell not toxic? And here we're really talking about such as the use of cancer therapeutics in cancers. They kill cancer cells, but we know they don't always work. And we're going to focus specifically here on the mitochondrial pathway of apoptosis. So again, I think you've all been introduced to this. I won't go into a lot of detail here, except to say that the mitochondrial pathway of apoptosis, or the intrinsic pathway, is induced by a variety of stressors, therapeutic agents, but we know lots of other things. They require the activity, it's regulated by the activity of the BCL2 proteins and is dependent on the so-called BCL2 effectors, Bax and Bac, to some extent Bach. These are the effectors that permeabilize the mitochondrial outer membrane to release cytochrome C and other mediators like SMAC and OMI. Cytochrome C activates APAF1 to form the apoptosome, and that in turn leads to caspase activation in the cell, and the cells undergo apoptosis. The apoptosome is an elegant structure. Cytochrome C is the only ligand that we know can activate the apoptosome. And it's only the holocytochrome C which resides in the mitochondrial intermembrane space. You have to get cytochrome C out of the mitochondria to activate apoptosis in this pathway. And just as an aside, we actually now have movies of the apoptosome in action at a deep molecular level. Here's the movie. Right. That's just for Gustavo. We can also see this in cells, and I'll let this movie come around. Hopefully it won't stop. I'll have to go back and do it again. This is Cytochrome C Gfp, a movie made many, many years ago by Josh Goldstein in the lab. As the cells engage apoptosis, cytochrome C does come out of the mitochondria prior to any indication of apoptosis occurring. this was actually, these sorts of movies were the movies that this was the imaging that really convinced the community that it is the release of cytochrome C from the mitochondrial intermembrane space that triggers the mitochondrial pathway of apoptosis. Now, we often report when we do experiments, and if you look in papers, you add something that induces apoptosis and we say, this is the percentage of the cells that died. And we're perfectly happy to see that, okay, we treated and 70% of the cells die, and then we do something and less cells die. But we don't pay a lot of attention to the cells that didn't die. And if you think about it, when we take a culture of one cell type, it's not, if we use a cell line, all the cells are genetically identical. They're all being treated with the same agent. And just some percentage of the cells die and other cells live. What's going on in the cells that live? Halime Kalkoven in the lab is investigating this, and it's an emerging area of research. When we think about, there we go. When we think about cancer, we often think about drug resistance. The cell has mutated something, and that makes the cell resistant to the drug. What we've started to realize, and something we should have realized long ago, from that type of simple experiment I just described, that when you treat a population of cells with an agent that induces apoptosis, some cells die and some cells don't. They're all genetically identical. Is something changing in the cells that survive? That process, instead of resistance, is now being called persistence. Non-mutational drug resistance is persistence. And this is thought to be really important because if the cells that they didn't mutate, they didn't survive because they had a mutation, they just survived because they didn't sufficiently engage cell death. Those cells survive, and they could then acquire mutations that would make them resistant, truly resistant at a genetic level. This concept of persistence is emerging, and we're starting to realize that persistence, being exposed to an agent but not dying, can change the properties of the cell. Now, we wanted to study this by engaging as pure a cell death mechanism as we could. And to do that, we used a combination of BH3 mimetics, basically inhibiting the active anti-apoptotic proteins in the cell so that they would engage mitochondrial outer membrane permeabilization and die, but in a way that we're not inducing a lot of other changes. So we're not adding a drug that's perturbing other things in the cell, and wanted to look at something that's really right at the core apoptotic machinery. So we treat with a combination of ABT737 that inhibits BCL-XL, BCL2, and BCLW, together with an MCL1 inhibitor, and we've used a couple of different ones, but I'll just show you with this one. And we treat cells. So here we're showing HCT116. This is a colorectal cancer line, but as I'll show you, this is generally true in all the cell lines that we've looked at. If we treat them with a limiting dose of this combination of BH3 mimetics, there are some cells that persist that didn't actually die in that round. If you use enough, you'll kill all the cells. But if you use kind of a slightly lower dose, there are cells that survive. And when you then grow those cells up, they're more resistant to the treatment with that combination of BH3 mimetics. Not forever. If you let them grow long enough, they become just as sensitive as they were originally. But initially, for at least a few days, they continue to have this persistent phenotype. And that's just shown here in terms of kinetics, that if you take these persister cells, or if you take cells that don't have backs and back, they have less response, apoptotic response, kinetically and overall to the treatment with the BH3 mimetics. But they're also now resistant to treatment with a range of chemotherapeutic compounds. So if we use tunicamycin or ER stress inducer, or MG132, which is a proteasome inhibitor, or chemotherapeutic drugs, these cells show less capacity to undergo apoptosis in response to these signals. So they survived, they didn't mutate, they were just in a different state than the other cells, than the original parental cells. Now, we're not the only ones who observed this. Here's an example of a paper that was published in Nature a couple of years ago, looking at these drug-tolerant persister cells in response to a wide variety of chemotherapeutic agents. And one of the things this group noticed was that the cells acquire a vulnerability. And that vulnerability reflects a sensitivity to the induction of another cell death modality of ferroptosis. Now, ferroptosis is very different from apoptosis. Ferroptosis is caused by the oxidation of polyunsaturated fatty acids in the cell's membranes. Lipid peroxidation occurs as a result of what's called the Fenton reaction. It's when hydrogen peroxide interacts with iron in the cell, and that can cause the formation of lipid peroxides. Lipid peroxides themselves, in the presence of oxygen, will cause more lipid peroxides, and that can be catastrophic for the cell. And so cells need mechanisms to detoxify lipid peroxides. And one of those mechanisms is the lipid peroxidase GPX4. GPX4 is a selenoprotein that converts lipid peroxides to lipid alcohols, and its function depends on glutathione. Glutathione is present in cells. It's produced metabolically from cysteine and glutamine. Glutathione reconstitutes GPX4 so that it can continue to detoxify lipid peroxides in the cells. And there's a drug called RSL3 that inhibits GPX4. Indeed, if you treat HCT116 cells with this inhibitor of GPX4, there's a little bit of increase in lipid peroxides that's detected by this probe. There's an increase in lipid peroxides. But if you chelate iron, that goes down. But if you take the persister cells that survived the induction of the mitochondrial pathway of apoptosis, now inhibition of GPX4 causes a great increase in lipid peroxides over the parental cells. And again, this is dependent on this Fenton reaction. If you chelate iron, that's prevented. Consistent with what the other groups have shown. And indeed, the parental cells are pretty resistant to the inhibition of GPX4, but the persister cells are very sensitive. Again, as this group described. So there are changes in the cells that persist after being exposed to these BH3 mimetics. we saw this with other cell lines as well, again, like other groups have described. So we interrogated the transcriptomes of the parental cells and the persistent cells. And what we found is a number of gene changes in the cells that have just been, they didn't die, but they were challenged with the BH3 mimetics. And among the changes were changes in metabolism that might reflect, that might account for some of that increased susceptibility to inhibition of GPX4. But curiously, and we'll come back to this in a minute, we also saw a lot of changes in genes associated with cell motility, cell adhesion, migration, and cytokine production. And that'll be important in a minute. But we also saw a lot of changes in genes that reflect the activation of what's called the integrated stress response. So the integrated stress response is the response to a wide variety of cellular stresses. So ER stress, amino acid deprivation, heme deprivation, or viral infection can all induce the integrated stress response. These are through different kinases in the cell. There are 4 of them. 4 different kinases can engage, can cause this integrated stress response. And what happens is that these kinases phosphorylate a translation factor called EIF2 alpha. When this translation factor is phosphorylated, it no longer can engage cap-dependent translation of proteins. But there are proteins that now get expressed. They're not expressed at the RNA level. These proteins are expressed at the translational level. A key example is a protein called ATF4, which is a transcription factor. Normally, ATF4 is not translated. It's transcribed all the time, but it's not translated. But when EIF, and the reason is that it has an open reading frame that is out of frame and won't allow protein translation when you have cap-dependent translation. But when you phosphorylate EIF2 alpha, it changes the start site for the translation, and now the protein is made. And this is a transcription factor that induces the expression of a wide variety of genes associated with this integrated stress response. Indeed, we see that the persister cells phosphorylate EIF2 alpha. And remember, we are not stressing these cells by inducing ER stress or the amino acid deprivation or other things that normally induce the integrated stress response. Here, we're only inhibiting the BCL2 proteins to promote mitochondrial outer membrane permeabilization. And we induce the expression of targets of ATF4, the same kinds of targets that would be activated if we intentionally induced, for example, an ER stress response. I have no idea why that was. We can also look at this a different way. We can make a Luciferase reporter where we fuse luciferase onto the that open reading frame of ATF4, and it only gets expressed when EIF2 alpha is phosphorylated. And there again, we can induce the expression of this indicator either by inducing ER stress or by inhibiting the BCL2 family members to induce mitochondrial outer membrane permeabilization. So we can see this in other cell lines, not just that one. We see this in a number of different cell lines. We have the persisters that can survive subsequent treatment. They have upregulated they're also resistant to a variety of other inducing agents, and they upregulate these targets of the integrated stress response. Same thing here with another cell line. So does this matter? Well, as I mentioned, ATF4 is activated. We also did some other genes that were activated. We silenced those. If you silence ATF4, this ability to persist in the face of this very specific stimulus goes away. ATF4 seems to be important for that ability of these cells to persist in the presence of BCL2 family inhibition. But here's where it gets really interesting. If we take persister cells, they actually grow quite slowly. The persister cells, and other people have described this as well, the cells that persist these stimuli for the induction of apoptosis don't grow very fast. And in fact, when we put them into animals, tumors grow initially more slowly. But then the persister cells take off and they invade and they metastasize to a much greater extent than the parental cell line. We see this in a number of our cell lines that we treat to induce apoptosis, but the cells that persist, although they divide more slowly, colonize and metastasize to a much greater extent. And here's where it's really interesting. That effect is not just due to the drugs and due to the inhibition of the BCL2 proteins. It's absolutely dependent on the presence of BAX in those treated cells. So the induction of MOMP, the activation of BAX are driving this phenotype of increased invasiveness and aggressiveness in the cancers. So persistence in the face of insult is not understood, but may represent stochastic states of transcriptional regulation or a compensatory response to the insult. Can we target susceptibilities that arise with this persistence in the treatment of cancer? A very active area of investigation, and I gave you some examples with the inhibition of RSL3. The transcriptional landscape of persister cells is associated with increased cancer aggressiveness and this integrated stress response. But this depends on BAX and may therefore depend on actually surviving mitochondrial outer membrane permeabilization. How does MOMP, mitochondrial outer membrane permeabilization, induce an integrated stress response? And is this actually responsible for the increased aggressiveness? Experiments are underway. They're really, really interesting. We think we have a unique pathway for the activation of the integrated stress response that we're investigating, but I'm going to leave that right there at the end of that riddle and see if there's any questions.
Doug, I have a quick question.
Yes.
In these very cool experiments that you are performing, do you have any idea if cytochrome C release from the mitochondria plays any role? If so, does it related to activation of apoptosome or production of ROS? And together with that, is ROS related to integrated stress response?
So we have looked at that. It does not appear that this is ROS mediated. Inhibition of ROS doesn't seem to prevent these effects. Inhibition of caspases also doesn't affect these changes. And we're doing the experiments right now to look at APAF1, but all of our indications are that this is APAF1 independent. There's two possibilities, and we're investigating both of them, and I'll say it's very preliminary, so I don't want to commit myself. but let's just say it's possible that it's cytochrome C. But we also have another candidate. So we'll know the results soon.
Cool. Very cool.
And we believe that we've identified the stress kinase that's responsible. I can tell you that it's not PERK. It's not the ER stress kinase. It's one of the others. But repeating the experiments and doing the appropriate knockouts.
Last question?
Sure.
Yeah, look, really interesting stuff. We're doing something quite similar, but looking at, you know, what cells, what kind of responses they make, particularly inflammatory responses when they don't die. But I just wondered about this issue of you saying it depends on backs and back. Is that particularly in this context because you're using BH3 mimetics?
Oh, it's quite possible, Seamus. It's quite possible. And we intentionally are using BH3 mimetics because we want to focus on this core pathway. But yes, absolutely. If we killed the cells with an ER stressor, I fully expect that we would engage the integrated stress response through PERK. Or if we used other drugs that perturb amino acid availability or engage PKR, certainly those could be involved. But we specifically wanted to look at what happens when you specifically engage the mitochondrial pathway.
Yeah. I'm just wondering if you also use something like a real world chemotherapeutic that does ultimately engage that pathway, but it does it in a much more kind of distal way.
Yes.
Will it still be back dependent, this persister, you know, phenotype?
I think it's a very good question. I don't know the answer. I think it's worth finding out. We're doing some of those experiments now with persistence in response to a broader chemotherapeutic. You've nicely shown that microtubule inhibitors and ER stressors induce cytokine responses. They, of course, will also induce integrated stress responses. I don't think that those are necessarily back dependent, but and also the target we do think is engaged is also not necessarily back dependent. We can engage it in a back independent manner. I can also mention there's one paper in the literature that says if you enforce the expression of ATF4, cancers become more aggressive. But we're interrogating that in the context of all of our systems. But of course, I don't think ATF4 is going to be the whole story. Certainly cytokine production is going to have a role, and there are other ways to engage those. But I think when we go to a general therapeutic, it's much more difficult to separate all of the different arms that might be contributing.
Yeah. And I'm just wondering if the, you know, when the cells take off and become much more aggressive, whether there's any DNA damage at that point. I mean, is, for example, it CAD dependent? Is it possible that you're getting a degree of MOMP and CAD activation? We're
also looking at the role of CAD in that phenomenon. I don't have an answer yet, but I'll come back to CAD later. Caspase activated DNAs and mutation, I'll come back to that a little later. Cool.
Thanks.
Next riddle. This is a short one. This is a short one, but I think it's one that is important and really opens up a lot of questions. So how dispensable is something that is essential? If it's essential, it can't be dispensable. And the reason I ask that is this statement is made at the beginning of almost every review article on apoptosis, and pretty much any other article on apoptosis. Apoptosis is essential for development and homeostasis, isn't it? In fact, I just did this. I just put in apoptosis is essential into Google, and I got over 1 million hits. we all think apoptosis is essential. And then this experiment was done. So again, I'm only going to talk about apoptotic pathways. Now, the mitochondrial pathway of apoptosis and the actual experiments that were done didn't relate to the death receptor pathway, but we'll come to the death receptor pathway right after this. The mitochondrial pathway of apoptosis has to be really important. This is the major pathway that's engaged, the major way you engage apoptosis. Apoptosis is the major way cells die in animals. It's dependent on Bax and to some extent Bach. And this paper came out. This is a paper from the laboratory of Andreas Strasser. They knocked out Bax, and Bach. And they saw, maybe not unexpectedly, that most of the animals did not survive embryogenesis. 95% of the animals did not survive embryogenesis. But 5% of the animals did. And when they looked at those animals, the tissue architecture of most of the tissues was perfectly fine. The livers were normal size, the kidneys were normal size, the gastrointestinal tract looked fine. Most tissues were perfectly fine. Hematopoietic tissues overexpanded. There were lots of developmental defects, but it wasn't absolutely essential. And if we go back to so much of the thinking about apoptosis and its role in development, of course, we always cite the role of apoptosis in digit formation, and there it might be really important. But it was absolutely required for development and homeostasis? The answer seemed to be no. It's really important for developmental timing events, but it may not be absolutely essential for maintaining the architecture of a tissue. And that's something that we're going to have to think a lot about. While apoptosis certainly contributes to normal development and homeostasis, it may be less essential in many tissues than we once thought. Our ideas of the essential role of cell death in sustaining tissue homeostasis in the face of cell division may have to be rethought. We've often said that in order to maintain the size of a tissue, cells are proliferating, but cells are also dying, and that needs to balance in order for that tissue to stay at its normal size. That doesn't seem to be true. And how's that going to affect our thinking about cancer, wound repair, and general physiology? Though experiments of the type that Andreas Strasser did are very difficult to do, and they even bred those mice to an MLKL knockout to take out necroptosis and didn't see any difference. It's not clear that cell death is absolutely required for development and homeostasis, although it's certainly very important. 95% of the animals didn't survive. Okay, but in thinking about development and thinking about the roles of cell death pathways in development, we can also think about the death receptor pathways and necroptosis. And here, just really quickly, going to show a tidbit because it's very mysterious of how complicated the integration of cell death pathways are. We think it's already pretty complicated, that caspase 8 can promote apoptosis, but prevents necroptosis. And this guy, Bart Tummers, wanted to go a little further into a basic observation that we made more than 10 years ago. It's been known for almost 25 years, over 20 years, that if you knock out caspase 8 or FAD, the adapter for caspase 8, animals don't go through development. And their failure to go through development is not because of a lack of apoptosis, but because the yolk sac doesn't properly vascularize at day 10.5. And 100% of these embryos die at day 10.5 in a caspase 8 knockout. If you cross these animals with animals that don't have RIP kinase 3 or MLKL, 100% of the animals survive perfectly well, and they grow up to be adults. So the role of caspase 8 and FAD is to prevent the function of RIP kinase 3 and MLKL. Well, what Bart wanted to do was look at that in a little more detail. So he made animals that carry mutations in caspase 8. He made a mouse where caspase 8 can bind to FAD, but it doesn't oligomerize. It'll form a dimer with flip, but it won't form a dimer with itself. He also made a mouse where caspase 8 can dimerize, but it doesn't autocleave. Both of those mice were perfectly fine developmentally, and they could grow up to be adults. And if he crossed those mice with an MLKL knockout, they grew up perfectly fine, and they could be adults. We also knew previously, as I just showed you, that a FAD MLKL double knockout develops perfectly fine, and they grow up to be adults. And then he made a cross of the FAD knockout, the caspase 8 knockout, the non-cleavable caspase 8, and the MLKL knockout. These animals were born, they would get through development, but they all die within 2 weeks. Here's what it looks like. All the animals die before 2 weeks with the non-cleavable caspase 8, FAD, MLKL, triple mutants. Now, importantly, if you knock out caspase 8, FAD, and MLKL, those mice grow up. They are developed and they grow to be adults. Of course, the FAD MLKL double knockout with the wild type caspase 8 develop fine and they grow up to be adults. These mice all die before 2 weeks. So first, this told us absolutely that caspase 8 can have a function that's completely independent of FAD. That hadn't been seen before. Caspase 8's only adapter molecule, as far as we knew, was FAD. Now we know there's another one. And if you looked in these mice, there was massive inflammation of their tissues. The skin, the gut, the lung, and also other tissues. They're dying of a massive inflammatory disease. So Bart knocked out caspase 1 in the background of these animals, a massive undertaking. He also knocked out RIP kinase 1. And more recently, not shown here, he has now knocked out ASK, the adapter for caspase 1. And knocking out RIP kinase 1, caspase 1, or ASK completely prevented this death within 2 weeks, and these animals all grow up to be adults. So all I can say about that is the basic observation: caspase 8, non-cleavable, MLKL deficient animals grow up. If you remove FAD, or if you remove RIP kinase 1, they're all dead by 2 weeks of life. If you take these animals that don't have RIPK1 and remove FAD, they'll grow up to be adults. If you take these animals and remove RIP kinase 1 or caspase 1 or ASK, we now know, they all grow up to be adults. I don't expect you to understand it because we regard this as a mind melt. The deletion of caspase 1, ASK, or RIP kinase 1 prevents pathology in animals that have a non-cleavable caspase 8 but don't have FAD or MLKL. Go figure. Something to figure out. And I'll stop there. There might be questions, but I probably won't be able to answer them, although we know a great deal more about that non-cleavable caspase 8.
I have a question, Doug.
Go for it, Gustavo.
So does this non-cleavable caspase 8 could form a platform for activation inflammasomes?
That's what we think is happening. And our evidence is, and of course, I could give a whole seminar on this. We see that in epithelial cells, caspase 8, the non-cleavable caspase 8, and ASC associate with each other and form specks that is not seen in the presence of FAD. So we think that ASC and FAD compete for caspase 8, especially in these conditions where caspase 8 is not cleavable. So that's where that is right now. It is not an NLRP3 inflammasome. There may be another NLR that's associating, but it'll take a long time to figure that out. And it's not in macrophages. This is a phenomenon that's occurring in epithelial cells in the gut and the lung.
But do you think there will be like a third element, not only ASC aggregation of caspase 8 and then maybe cleaving caspase 1, whatever, or
maybe possibly, you know, I'll go a step further, and this is embarrassing because we should know the answer to this. So caspase 8, caspase 9, caspase 2 are not activated by cleavage.
Yeah.
So we use that a lot to say that it's activated, but cleaving those doesn't activate them. But we don't know that for caspase 1. And it's possible now that the cleavage of caspase 1 might activate it. It hasn't been tested. And it's embarrassing that nobody's done that yet. But we don't know the answer. It's also possible that caspase 8 is stabilizing ASC to recruit caspase 1.
Yeah.
And that's the other possibility.
And Doug.
Yeah.
And how about gazanine D or E in this model?
Oh, let's come to that next.
Cool.
So, next. The riddle starts with this riddle, but the answer is going to take us in another direction that should be fun and interesting. So if a cell dies in the forest of the body, does it make a sound? Do dead cells matter? And does it matter how a cell dies? Does it matter how a cell dies? So this is a project done by Caitlin Liu, and who's a cell biologist and immunologist, and Mark Chen, who's a computational biologist. And here we're going to talk about gasdermins. So as Karina knows very well and just asked, there are molecules in cells called gasdermins. We'll focus mainly on gasdermin E, but also gasdermin D in another form. Gasdermin D is cleaved by caspase 1 and also by caspases 4, 5, and 11. Gasdermin E is cleaved by caspase 3 and caspase 7. And in each case, the cleavage of the gasdermin causes it to change its conformation so that it now assembles into an elegant pore structure in the plasma membrane and possibly other membranes. And the cell dies by pyroptosis. It's a little more complicated. Again, gasdermin E is cleaved by caspases 3 and 7. gasdermin D is cleaved by caspase 1 and other caspases, but it's also cleaved by caspase 3, but the cleavage of caspase 3 inactivates it. So Caitlin generated a gasdermin D where she mutated that caspase 3 cleavage site, so caspase 3 wouldn't inactivate it. And then she mutated the caspase 1 cleavage site so that caspase 3 could activate it. So this mutated gasdermin D is a control for our gasdermin E to tell us that it's not, what we're seeing is not something unique to gasdermin E, that it's more about the pore-forming protein. Now, when a cell would engage caspase 3, if it doesn't have gasdermin E or it doesn't have our mutant gasdermin D, it'll undergo apoptosis. But if it has gasdermin E, it'll undergo a pyroptosis-like death. And that looks like this. So here are cells dying by apoptosis. And here are cells that express the mutant gasdermin D, and they don't die anymore by apoptosis. They just die in this sort of necrotic death. And the same, exactly the same thing is seen if you enforce the expression of gasdermin E. And that affects the ability of these cells to grow as tumors. So if we take B16 melanoma, which does not express a gasdermin, and enforce the expression of gasdermin E or this mutant gasdermin D, and I'm not showing you the control, the wild type gasdermin D is just like the parental. The parental tumor grows in mice as flank tumors really well, but these really don't. But if you put these cells into an animal that doesn't have an intact immune system, an NSG mouse that doesn't have lymphocytes or NK cells, now, if anything, the cells expressing the gasdermins grow a little faster. And in keeping with that, the presence, here it's just showing with the mutant gasdermin D, but the same is true for the gasdermin E. There's an increase in NK cells and T cells in these tumors. And in fact, if you deplete the animals of CD8 T cells with an immunoablating antibody, or NK cells with a NK1.1 ablating antibody, now those indolent tumors that express the gasdermin grow much faster. And this is really the induction of immune response, because if you remove the dendritic cells that will cross-present tumor antigens that depend on the transcription factor BATF3, the BATF3 knockout mice, the tumors don't grow in the wild type animals, but they grow in the animals that can't cross-present tumor antigens. So all of this would say that this pyroptotic death is inducing immunity, and that's what's preventing the growth of the tumor. And in fact, last year, this is just showing pooled data that our gasdermin producing tumors don't kill animals and the wild type tumors do. Last year, 2 groups published basically this, that gasdermin E suppresses tumor growth by activating anti-tumor immunity. So we wanted to check that in our system. We got scooped, but that happens. So we looked in the system, and indeed, our animals live. As I showed you, they induce immune responses. But we took the animals that survived and we gave them the tumor again. And they do show some increased survival. They live a little longer because they have this increased immune response, but they're fully susceptible to the tumor. They're all dying from the tumor. Whereas in the primary response, when they express the gasdermin D, these are the wild type tumors that don't have the gasdermin, the mutant gasdermin or the gasdermin E. These are animals that have survived the gasdermin E expressing tumor. They still die in response to the parental tumor. The anti-tumor immune response that's being induced doesn't seem to be enough to eliminate the tumor. So maybe we're missing something. So Caitlin used this system. If you take a tumor cell and express a probe called GCamp6, it's a calcium indicator. So this is a fluorescent protein that lights up when calcium levels increase. And if you expose these to a cytotoxic cell, as the cytotoxic cell contacts and gives a hit to the target cell lights up. And after a number of those hits, the target cell dies by that cytotoxic insult. Now, if she takes parental cells that express this GCamp and looks at calcium signal, the cytotoxic cells are inducing lots of calcium hits in these individual cells. And once it lyses, all the fluorescence goes away, so that cell's now dead, that cell's now dead, that cell's now dead. But you can see it took a lot of hits for that cell to die. Now you take the same cells and express gasdermin E, and as soon as the calcium goes up, they die. And we can quantify that it takes many fewer hits of a cytotoxic cell to kill a cell that has gasdermin E. And again, that's shown here. They die faster and they die upon a contact. Fewer contacts from the cytotoxic cells will induce this death, this cytotoxic death. And actually, we can even see this in a movie. In this movie, the red cells express the caspase-3 activated gasdermin, and the blue cells are the parental. And the cells that are unstained, that don't have a fluorescent protein, are cytotoxic lymphocytes. So here's the movie, let's see. And they're getting attacked, they're going after them. And you can see that the red cells are disappearing and dying. And the blue cells are even dividing. It takes much more to get them to die. Oh, there we go. So that would suggest that if gasdermin E is promoting sensitivity to being killed by an immune response, then if a cell loses the gasdermin, it can evade it. It might have a better chance of surviving. So Caitlin introduced B16 melanoma with gasdermin, with a fluorescent GFP. She took those tumors, injected them intravenously, and sometime later looked in the lungs at the colonies that formed. Now, if these cells just express GFP, the colonies all have GFP. If the cells express the gasdermin E GFP, there's a lot fewer colonies, but the ones that grow have pretty much lost GFP, meaning they've also lost the gasdermin E. If she does this in mice that don't have an immune system, they're all GFP positive, all of these metastases. If she takes a wild type mouse but ablates NK cells or CD8 T cells, now lots of metastases and they have GFP. So the immune system is selecting against the gasdermin E. But the gasdermin E is also making the cells more sensitive to the immune response. Now, indeed, many tumor cells silence gasdermin E. That's well described in the literature, and it's well described in the literature that tumors express varying levels of gasdermin E. Different tumors express different levels of gasdermin E. Mark actually did the correlation, and indeed it is the methylation of the gasdermin E promoter that's associated with less expression of gasdermin E. So tumors suppress the expression of gasdermin E by promoter methylation. But here's where it gets really intriguing. Across the entire cancer genome database, the TCGA cancer database, if Mark compares the number of tumor mutations to the level of gasdermin E expression across all tumors in the database, more than 8,000 tumors, there's a highly significant inverse correlation between how much mutation a cancer cell acquires and how much gasdermin E it expresses. The more gasdermin E that's expressed, the less mutations. And remember, it's not the mutation that's causing the loss of gasdermin E, it's methylation of the promoter. So somehow, there's a relationship between tumor mutational burden and gasdermin E expression. And actually, if we interrogate this relationship, it's really just a few, it's certain tumor types show this inverse correlation quite strongly. For example, head and neck squamous cell carcinomas, stomach adenocarcinomas, very high p-value in this inverse correlation, and a number of others, but not all tumors show this effect. Brain cancers really don't show this effect. Now, as I mentioned, there's other gasdermins. When Mark looks at the other gasdermins, this is gasdermin E here, also called DNFA5, DEFNA5. That has the very strong inverse correlation. None of the other gasdermins show this inverse correlation. None of the other poriform gasdermins show this inverse correlation with tumor mutational burden. Now, tumors also, some tumor types can be classified in terms of how much immune response is going on in those tumors. And it's been widely documented that tumors that are immunologically quiet have fewer mutational burden, fewer neoantigens. And people have suggested that that's why you need a lot of mutations to produce the antigens to induce an immune response. But I'm going to suggest that maybe it's the other way around. Using tumors that show variation in their types of immune responses, if we look at that relationship between gasdermin E and mutational burden, that those tumors that have immune responses associated with them show this strong negative correlation, whereas tumors that don't have immune responses of the same class don't show that inverse correlation. So what do we think is going on? And the answer may come from the work Stephen Tate has done and published on the induction of apoptosis and its consequences for mutation. What Stephen's described is that cells can engage a stress and can engage the mitochondrial pathway of apoptosis. But if only some of the mitochondria permeabilize, that cell can survive, but becomes genetically unstable. And here's why we think that happens. That as executioner caspases are activated, they cleave ICAD in the ICAD-CAD complex, which activates the caspase activated DNAs that produces DNA damage. And if that cell survives, it will have increased mutation. Now, when a cytotoxic cell engages a target cell, granzyme B released from that cytotoxic cell into the cytosol of the target cell activates caspase 3 and can activate this same process. If the cell survives, it may have increased mutation. Now we're starting to do those key experiments. Oh, and sorry, there's more. The hypothesis is that if the cell expresses gasdermin E, now the activation of the caspase may ensure that that cell dies rather than living with the DNA damage. So in other words, the gasdermin E may be a kind of thermostat that the levels of gasdermin E decide whether a cell that's under immune attack will die this lytic death at high levels of gasdermin E, or at low levels of a gasdermin E may survive with DNA damage. So is that even feasible? And here's how we're going after it. This is one of the approaches. An assay for mutation in cells is looking for mutation in an enzyme called HPRT. If cells express HPRT, they are sensitive to a toxic analog of guanine. If they mutate HPRT, they're resistant. So Caitlin took cells with or without gasdermin E. She induced the activation of caspases. She hasn't done it yet with the cytotoxic cells, but using 3 different approaches, BH3 mimetics, a drug called raptinol, or engaging the trail receptors on the cell surface. She allowed those cells to recover and then asked whether they became resistant to this guanine derivative that's only toxic in cells that have intact HPRT. If we focus here, cells, wild type cells that were treated with, I'm sorry, I can't actually see my, yeah, I can see it, that are treated with trail. Wild type cells that survived, more mutants appeared in the surviving cells that didn't have gasdermin E than did have gasdermin E. And these were using the same numbers of surviving cells treated with the drug. Same true with raptinol, and the same was true with the BH3 mimetics. Now we want to know if this will also be true under cytotoxic attack. And most importantly, we want to know if this will happen in vivo with or without an immune response. So it matters how cells die and survive from cell death. The mode of cell death can affect immune responses and mutagenesis. This potentially has direct implications for the relationships between immunity and cancer progression. I want to suggest that at least in some cancer types, the high mutational burden isn't what's driving the immune response. It's the immune response that's driving the high mutational burden by engaging this caspase-activated DNAs. Now, of course, to test this, we're doing the experiments with CAD and ICAD knockouts. We're looking at the relationships between the expression of CAD and these mutagenic phenotypes, and especially looking in vivo at cells that express or don't express gasdermin E, either naturally or by enforced expression, and how that affects the acquisition of mutations in the presence of an immune response. But as you can see, this has a lot of consequences for our understanding of anti-cancer immunity. Rather than stop there for questions, I'm going to go into just one last riddle because it's a very short one. Distressed doctors document directed deaths by dastardly deeds in droves. How many dastardly deeds can documenting doctors dictate? That's the riddle. What it really is how many kinds of cell death are there? Some reviews show this number of cell deaths. Another review showed this number of cell deaths with when they were originally described, although there's a typo on this reviews because necrosis was not described in 1965, it was described in 1865. But as you can see, many types of cell death are described and continue to be described. And often people ask, how many kinds of cell death are there? I want to suggest that maybe we start thinking about types of cell death in a slightly different way. That there's cell death that's effectively murder. There's cell death where the damage to the cell kills the cell directly. It cannot survive the damage. There are forms of cell death that are what we could call cellular suicide, where the cell effectively kills itself. And it kills itself because it engages a pathway that is probably evolutionarily derived, that this is a pathway that occurred in evolution and conserved in evolution, and that pathway is effectively a cell killing itself by suicide. But then I think many of the types of cell death that are described and continue to be described are effectively sabotage. These are still active cell death or regulated cell death, and I would include here ferroptosis and parthenotose and lysosomal cell death, that these are forms of cell death that occur and are dependent on cell activity. But the reason I call them sabotage is because if you think about sabotaging a moving train by removing a railroad tie, if the train isn't running, nothing will happen to the train. But if you remove the tracks and the train is running, the train will crash. And that's effectively what we're doing when we're describing many of these types of cell death. We're poisoning an enzyme, we're poisoning a mechanism in the cell, and that cell dies. We've sabotaged it. It doesn't mean it's not important, but it's a different way of thinking about it. The pathways that have been, that have arisen in evolution and are conserved in evolution may be distinct from these sorts of sabotage pathways. They may be actual cellular suicides, and maybe those distinctions can be a little more useful in separating types of cell death pathways. So I'm going to stop there, except to say cell death is a fundamental cellular process, and we have a huge amount to learn. I predict over the next 10 years, and I'm even predicting over the next couple of years, with the stories we're trying to develop, but also the stories that are being developed by all of the people who presented in your course. We're going to learn a great deal more, and this is a vibrant, ongoing field of investigation that is far from complete. And now I'll take questions about the last part or about any part. But let me also just give a little acknowledgement. I talked about some of the Green Lab alums, Andrew Oberst and Yinan Gong and Stephen Tate. Other alums have spoken today as well, but I won't embarrass them. I also talked about current work being done by Halime, Caitlin, Bart Tummers, and they're also being helped by other people in the lab. I should have put Mark Chen on here as well. Sorry, left him off, and a number of people who have helped. And finally, I really have to give a shout out to somebody who Gustavo knows really well, and Seamus knows well, and Christina Munoz knows well, Pat Fitzgerald, who retired a few months ago. He's waiting to move to San Diego. When things are over, there's Pat. Well done. And we couldn't have gotten where we are without his amazing help. So I'll stop there. And if you have any questions on the last part or about any of the parts, let's open it up.
Doug, I'm breathless. You know, it's such an amazing lecture. It's just incredible. I have a few questions. Seamus is showing some picture. I'm happy that it's not gonna appear in the YouTube only for us. It will appear though in the YouTube if Doug turns off the screen. Then I think yeah, YouTube will be, you know, listen to seeing.
Oh, and I get to say hi to Ricardo. Hello, Ricardo.
So
great to see you.
There are a few questions in the YouTube. I'm trying to put together my mind here to see a logical way. Before that, I just want to ask one thing. Your last part, when you mentioned also the work of Steve Tate, it's a bit reminiscent with you know, one of your previous riddle that we talked about, and I asked about cytochrome C or whatever it means. So can you try to combine this riddle in one, or is very different, or you think is different?
Well, they're all interrelated, of course, and the riddles are just made up, right? But yes, this all relates to cells that engage a cell death pathway but survive, and what the consequences of that could be. And here, in this last part, I want to suggest that the gasdermins, especially gasdermin E, is really important for setting the thermostat for how much caspase activity can you have in a cell without the cell dying. So as we know, the caspases, caspase 3 and 7 act on, our example is mutation, act on caspase activated DNAs to promote DNA damage. If the gasdermin E thermostat is turned up, the amount of caspase activation you need for the cell to die by activating the gasdermin is lower, and therefore the probability that that cell will survive with the consequences of CAD activation is decreased. And I think that's the reason why we see this remarkable inverse correlation between the level of gasdermin E and the mutational burden. I don't think it's only cytotoxic cells that are doing that. I think any activation of the mitochondrial pathway or any caspase activation in the cell, if that cell survives, it may be surviving with more DNA damage. And that would have to be entirely dependent on caspase activated DNAs. And that's going to look really interesting as we start to examine how important is the caspase activated DNAs for the acquisition of mutation in cancer.
Right. This comes to a question that I asked Seamus in the first talk, and this is related a bit with old experiments by Joe Trapani and others with granzyme B and A and perforin deficient mice. So what's the status of CTL or NK killing? You know, what are the molecules? Because in this context, apparently granzyme B plays a crucial role by activating caspases. But those old experiments would suggest that you know, perforin would be more important than granzyme or things like that. What do you make of it?
No, and absolutely. Of course, granzyme B is not the only granzyme that's important for killing. We also know cytotoxic cells don't even need perforin necessarily. They can kill by death ligands, by fast ligand or trail. But it's an interesting question, and we're doing those experiments. So one of the experiments that we're doing is to see, so remember the experiment I showed that the cells expressing the gasdermin E GFP lose the gasdermin E if the immune response is intact. So we're doing that experiment now. We've gotten the granzyme B knockout mice to see whether that's really granzyme B dependent, or the extent to which that's granzyme B dependent. I can also tell you, there's a nice paper from Feng Xiao that shows that gasdermin B is a target of granzyme A. Sorry, the letters get really confusing, but so granzyme A, when it's introduced into cells, induces necrosis. And that's been shown for many years, and there are all these models. He showed, no, those are all wrong. It's really gasdermin B is the target. And gasdermin B is cleaved by granzyme A, and the cell dies. We've done the experiments, and first of all, the mutational burden correlation does not hold for gasdermin B. It doesn't make any difference. And we've also put gasdermin B into target cells, and somewhat surprisingly to me, that did not sensitize them to cytotoxic cells. The tumors did not grow more slowly. In fact, they grow a little faster. So.
Okay, so let's start with some questions at the YouTube. Karina, you want to?
Yes. First of all, fantastic, as you used to say, fantastic closing lecture. So thank you, thank you, thank you. One question is from April Darcy. Do you think Apoptosis induction would be a good way to eliminate persistent cells, attack on all fronts?
It's a
and
really,
in
yes.
in the same line, do persistent cells have inflammatory phenotypes? What kind of contribution do they have to the TME?
It's a very good question. These are great questions. We do see an increase in expression of certain cytokines from persistent cells. It's made a little bit complicated, and I don't want to go into somebody else's work, but it's not all of those inflammatory changes are dependent on Bax and Back. And this is, goes back to a question Seamus asked. But they're not, in this case, we're not talking about inflammatory cytokines. We're talking about another class of cytokines. And that's not Bax-Back dependent. And that is work Stephen Tate's doing, and I'm sure we'll publish a nice story on, but I shouldn't talk about it. The question about gasdermin E and persister cells, yes, I absolutely think that gasdermin E levels will affect persistence. That's certainly what our data says. I don't know, though, yet how you can induce the expression of gasdermin E in a cell that isn't expressing it. I think it's very likely that if you enforce the expression of gasdermin E, you would have some effect on persistence. though, as we saw, expression of gasdermin E by itself does not limit the ability of a tumor to grow in the absence of an immune response. So they grow just fine. But yes, it would vary. We would predict, and indeed we've seen, that it prevents the appearance of persisters. I just don't know how to do that practically.
So Doug, in a follow-up question there, have you watched this also in other types of cell death? You know by Ina's work, right? You did with Ina, and that you can make undead cells from necroptotic signaling. Would you expect that these cells also persist and have this trends toward accumulating mutations and being more aggressive tumors?
Yeah, it's a great question, and it isn't one we've interrogated yet. I like the idea. If caspase-activated DNase is responsible for the increased mutation, and that's something we're testing, then my prediction would be that no, survival from necroptosis wouldn't promote mutation unless there's another mechanism, which is always possible, and I think it's really worth doing. It's a great question, and as you know, Ricardo, these are things that happen when people leave the lab. I asked Yinan if those are experiments that he was happy with us pursuing, or was this something he wanted to pursue in his lab? And he said he wanted to do it. So we'll wait to see what he tells us in terms of whether resuscitated cells change their properties as tumors.
So Doug, just to follow up as well, do you consider that the DFF would not be related to this integrated stress response? So you think you are trying to find like one really element, and why not like this integrated stress? You have like oxidation of DNAs and a lot of things that may contribute. Not only,
Well, we're doing the experiments. So we're looking at the relationships between caspase-activated DNase and the integrated stress response. The cells are ready, we just don't have the results yet. And to see how important that might be to the in vivo phenotype. It's possible that so the integrated stress response doesn't induce oxidation. If anything, it reduces it. It limits mitochondrial activity and a number of other effects that should reduce reactive oxygen. One of the things we've seen, though, in persister cells, they upregulate NRF2, which is strongly antioxidant. But that doesn't seem to matter, at least for the effects we're seeing. So, and I don't know how universal that is. There may be other explanations for that. And it might be fun to follow that up in the future. I've got a few ideas about how NRF2 is being activated, but the whole relationship between reactive oxygen and apoptosis, I think, is still really messy. We really don't know how important reactive oxygen is to the process of apoptosis. We certainly know apoptosis can proceed in the absence of reactive oxygen, but that it's engaged, and can it have any of the effects downstream? It's possible. But I know, at least from Stephen Tate's work and his published work, that the increased mutation that you see with the induction survival from apoptotic insults is completely dependent on caspase-activated DNase and not reactive oxygen-induced DNA damage. Yeah. So I suspect we'll find the same thing for our cytotoxic cells. Seamus.
Yeah, great talk, Doug. Lots of provoking ideas there.
That's the
So,
whole point.
Yes. I just wanted to ask about the Gasdermany story. So there's a few interpretations of that. Couldn't it also be that there's a kind of immune editing going on there, where if you have Gasdermany, you're basically eliminated at an earlier stage in, let's say, of the tumor progression. And so you get to live longer as a tumor cell if you have low Gasdermany, so you get to accumulate more mutations.
Well, I would say yes, Seamus, except, this is across the TCGA, and these are all tumors that have been resected from patients. So it's not a cell line experiment. These are sequences of, this is all data that's collected from straight RNA-seq and DNA sequencing. So yes, I get what you're saying, and that would be compatible, but if it was only cell lines. But this is in actual tumors.
Yeah, no, I mean, I know that, but it's still, I mean, for example, then you'd expect to see patients that had low Gasdermany, presumably they have a much poorer survival rate.
Well, that's actually described, yes.
So that's known.
Yes.
Okay.
And Gasdermany is actually described as a tumor suppressor on that basis. So that if you look across tumors lose Gasdermany, they suppress its expression by DNA methylation, by promoter methylation. If you don't have Gasdermany, prognosis is worse. All of those things are in the literature, yeah. But why that is only suggested from these two recent Nature papers that suggest the reason is because Gasdermany is promoting anti-tumor immunity. And we've kind of turned that on its head. Well, yes, you do get more anti-tumor immunity. And yes, the tumors that express Gasdermany grow in an immunodeficient environment, but not an immune attack environment. The actual reason we would suggest is that because Gasdermany is making the cells more sensitive to the immune response, not because they're inducing a better immune response. They do induce a better immune response, but that immune response cannot account for the effectiveness of the anti-cancer response.
So again, if this is the fact that these cells you think are getting multiple attacks, they don't have E, so they don't die. So then what about BID and caspase as being substrates? I mean, is Gasdermany a much, much better substrate for granzyme B, for example, than BID or caspase-3?
Well, you're the one who taught me that caspase-3 is not a very good substrate for caspase-3.
For granzyme B.
For granzyme B, but also, I see. You're saying so you're active, so granzyme B is engaging BID, it's engaging caspase-3, and it's engaging, and through this, it's engaging Gasdermany. There is some literature that says granzyme B can cleave and activate Gasdermany. We cannot reproduce that. We don't see any Gasdermany cleavage by granzyme B. So we would suggest that it really is acting as a thermostat for how much caspase-3 is being activated. Now, yes, you're right. BID cleavage will occur. That can also promote the death of the cell. And yet we also know that these cells very often are surviving. I mean, they can survive multiple hits by a cytotoxic cell and not die, right? So really the question is just going to be, and it's all based, it's all mostly computational right now. The real question is going to be, if you take the cells that survive insult by cytotoxic cells, do they increase mutation in a CAD-dependent manner? And those are the experiments that we're doing right now, just to test that. Yeah. I think if it fails, your explanation may be why.
Doug, so there's a question by Cecilia Almeida. It relates to the first part of your talk. She would like to know, I will combine her question here. So what size are the necroptotic corpses compared to the apoptotic corpses? And could it be considered like a particle, a vesicle that could be used for cell communication? Like may they have an effect outside the site of the dead, the tissue?
Oh,
like that?
Absolutely yes. So there's two questions there. So the cell corpse in apoptosis, it tends to shrink unless gasdermin E is engaged. In necroptosis, the cells swell. So the size of the corpses are different. But in both cases, there's a lot of production of extracellular vesicles. So in apoptosis, there's production of extracellular vesicles. I don't want to call them exosomes because they're really not, but they're extracellular vesicles. And Chris Gregory in Edinburgh is studying those quite extensively. And then in necroptosis, there's a production of extracellular vesicles. And Mordecai Gerlich in Israel is studying those. And both groups would absolutely suggest that these can act as vehicles for intercellular communication with the dead cell. And the interesting thing is that these, at least in the case of necroptosis, but perhaps also, I think it's also possible in the case of apoptosis, that these extracellular vesicles may be produced even if the cell doesn't go on to die. And that could be a very interesting area of research as well.
Okay. Ka, do you have another question from the YouTube?
Karina, you're muted.
I am muted. Sorry. We have, I think, the last one from Bruno Pires. So thanks for your great talk, Dr. Green. My question is related to cancer research in general. We are always looking for inducing cell death, but I wonder if we are underestimating other effects such as senescence or agonescence, which are critical for the metastatic disease, for instance.
Absolutely yes. And in a sense, let's go beyond senescence. We want to know about cytostasis, or maybe that's what you said. We want to know about dedifferentiation, as well as cell death. All of these things are highly relevant, but the people here who are speaking today are cell death researchers. We can't study everything at once, but the community certainly can. And there's a great deal to learn from each other in terms of relationships between cell death and senescence. How do some of the cell death pathways contribute to the senescence-associated secretory patterns? What are the interrelationships? How does the cell decide to senesce or die? And what are the different consequences of that? But let's face it, we can only study so much. So I would encourage you, Bruno, be the pioneer who finds the interrelationships in the context of cancer therapy, and that'll make a great contribution. for sure.
Doug, I almost forgot the question from Marcela Bitar, and she would kill me. She's a new MSc student in my lab. So she's asking, you show situation when the persistent cells occur in vitro and in condition of tumors in cancer. What about other physiological situation where we have a lot of like tissue turnover or infections or injuries? So do you see those cells appearing as well?
I think it's a great question. And I'll tell you what we're struggling with. And hopefully we'll get clever and come up with a way that works. But what we'd like to do is have a way to mark persisters. So imagine some magical system. We have if this could work, imagine a Cree which is only activated by caspase. And you can have a cell that will turn on that Cree but not die. And now that Cree will induce the allow the expression of a fluorescent protein. So now you can see the cells that didn't die have turned green. Now imagine if we make that animal, or if we make that tumor, and now be able to trace, okay, these are the persister cells, and this is what they're doing. So wouldn't it be amazing if, for example, we had that probe and it worked, which it doesn't, but eventually we'll figure out how to make it work. Imagine we had that probe and all the right controls, and then we saw, oh, look, when we damage this tissue, when we damage this organ, we see a bunch of cells turning green, and those are the cells that are repopulating the stem cell pool, or whatever that might be. We would learn a great deal about this relationship between, because don't forget, this phenomenon that we're seeing in the tumor cells, that's not something tumor cells invented.
Right.
That's some kind of fundamental process that probably plays roles in normal physiology. But until we really understand it, we don't even know where to look. Right?
So yeah, imagine that the designing animal with time would be all fluorescent.
Maybe that, it's quite possible. But we need to figure out more sensitive probes than we have. Right now, the probes are just not sensitive enough to really mark the persister cells. But I think we're going to start to understand how these things are fitting together in terms of physiology as well. Cancer models are just, in some ways, an easier way for us to probe that biology, but also highly relevant to disease.
So regarding the DA, the caspase DA story, Gustavo mentioned that maybe the caspase can directly be a platform for ASC activation and caspase-1 cleavage. But then there is RIPK1 knockout there, right? Which
Yes.
wouldn't explain there.
No, somehow RIPK1 is playing a role. Of course, we don't know it's because it's in the complex. It could relate to expression of a gene. These are knockouts. So these are genetic experiments. And I don't have any really good explanations for this. Also, why would it be tissue specific? So we don't see this. If it was just ASC binding to caspase 8, and that's now forming a platform, why wouldn't that be true in macrophages? We don't see it. We only see these aspects appearing in epithelium.
Do you think that RIP1 is required to transcriptionally activate the inflammasome genes and upregulate these pathways? Then you can have the full ASC platform formed?
Possibly. I mean, the trouble is, we need to figure out a simpler genetic model that recapitulates this, because right now it's a triple mutant that we make a quadruple mutant, and it's just horrible. Maybe as CRISPR techniques become better and we can generate these quadruple mutants directly, maybe we can learn a lot. Right now, each mutant takes, 2 years. And Bart started these, you can imagine he started these 5 years ago. But amazingly, he did an incredible job on the genetics, and he was able to make quadruple mutants that survive when all of their parents, all of the genotypes would be dead. These survive, and it's incredible. Yeah, in that setting. So that paper, you probably know, was published last year, and there's a lot of other data in there about it. But yes, it's quite mysterious. By the way, that paper also showed Seamus's fatosome working in vivo, which we were really, really happy about. And direct application of, we would not have figured that out without Seamus's work on the fatosome. No way. There were interesting phenotypes like the caspase 8 non-cleavable. If you inject those mice with Fas ligand, they survive because they don't fully activate caspase 8. If you cross them to the MLKL knockout, the mice are fine, but now if you inject them with Fas ligand, they die even faster than a wild type mouse. And why in the world? And we could show, though, that cell death was completely different. It was inflammatory cell death. It was inflammatory animal death. It was a cytokine storm, and it was dependent on the fatosome. Yeah.
All right. So
just giving Seamus the shout out there.
That's nice. Thanks.
No, it's true. It's totally true.
Cool.
All right, Doug.
People must be exhausted. A whole day of cell death talks.
The whole week.
Whole week of cell death talks. So everybody should come to Virtually Dead meeting now that you know so much about cell death. There will be the amazing Virtually Dead meeting, and this time on inflammasomes.
Yeah, I mentioned to them briefly, but I will send everybody, a note about the Stephen and Anna's organization of these workshops, and hopefully, they will join as well.
Oh, and by the way, last thing I want to mention is if you really find this whole study of cell death interesting, and you want to know more about it, maybe even work in this field, there's a really good book.
Second edition now released.
Yep.
Yeah,
you can get it on Amazon.
Amazon, cool. Yeah, I have it.
It sold hundreds of copies. Dozens of copies.
I got my personal one signed by the author, so that was very special.
That makes it worth less.
Yeah, probably. Yeah. Can I get one? Can I get one that isn't written in? Yeah.
Yeah.
Actually, you can get signed copies online too, I think.
You know the line from Malling Hill? Signed by the author, yeah, I know. If you can find an unsigned copy, it's worth a fortune.
That's right.
Shame is always funny.
Yeah.
Okay, guys, I have to break down now the transmission to the YouTube. Super thanks. If people want to give us a feedback, you have Karina, Ricardo, and my email. We would love to hear from you. And what about that, Seamus? Well, that was age. You look like my son now. Looks like Gabrielle. No, this was ages ago. Anyway.
That's great.
Okay, so goodbye to the audience in the YouTube. Thank you very much.